Without a constant input of energy, biological systems rapidly decay into thermodynamic equilibrium, in other words, death. This A2 unit is all about how cells transfer energy from organic molecules into adenosine triphosphate, or ATP. ATP is the universal energy currency of the cell. You will see exactly how it powers active transport, muscle contraction, and anabolic reactions, and why its precise biochemistry is heavily tested in Paper 4. To understand respiration, you must know exactly where it happens. Cambridge examiners actively discourage unscientific cliches like the powerhouse of the cell. Instead, A-star candidates define the mitochondrion as the exact site of aerobic respiration and ATP synthesis. You will map its highly folded inner membrane, the cristae, and the fluid matrix, linking these specific structures directly to their biochemical pathways.
The journey of aerobic respiration is a sequence of interconnected stages. It begins in the cytoplasm with glycolysis. You will track how one 6-carbon glucose molecule is split into two 3-carbon pyruvate molecules, yielding a small net gain of ATP. Then, in the link reaction, pyruvate enters the mitochondrial matrix, undergoing oxidative decarboxylation to prepare for the next stage.
Once inside the matrix, we enter the central engine of aerobic respiration. Here, you will not just memorize a circle of names. You will track every carbon atom lost as carbon dioxide and every hydrogen atom removed. These hydrogen atoms are picked up by coenzymes, NAD and FAD, which act like hydrogen taxis, transporting them to the inner mitochondrial membrane. Those hydrogen taxis deliver their cargo to the grand finale of respiration. At the inner membrane, the electron transport chain uses the energy from those hydrogen atoms to pump protons, creating a steep gradient. You will learn how a process called chemiosmosis drives protons back through ATP synthase, spinning the enzyme to generate vast amounts of ATP, with oxygen acting as the final electron acceptor.
But what happens to a cell when oxygen is completely unavailable? You will compare the anaerobic pathways in mammals and yeast. You will see how cells reduce pyruvate to lactate or ethanol just to regenerate NAD, keeping glycolysis running so they can survive, even though the ATP yield drops dramatically.
Finally, we look at the fuels themselves and how we measure them in the lab. Why do lipids release twice as much energy per gram as carbohydrates? You will calculate the respiratory quotient, or RQ, by dividing the volume of carbon dioxide produced by the volume of oxygen consumed. This simple ratio lets you deduce exactly which substrate an organism is metabolizing inside a respirometer, a classic Paper 5 scenario. By the end of this unit, you will be able to track carbon and hydrogen through the 4 stages of aerobic respiration, explain the precise mechanism of chemiosmosis, and calculate RQ values from experimental data. Let's get started with our first topic, energy and ATP.